Dear student, good morning to all of you. This class is M.Sc final year Inorganic special paper 7th A. Our topic will be continued supramolecular chemistry. Today we discussion on supramolecular reactivity and catalysis. What is the reactivity? What is the reactive of supramolecule and what is the importance of in catalysis? You know that catalyst can be complete the reaction in very fast and slow. This process is known as catalysis. In this process, what is the importance of supramolecule? We discuss in this topic. The design of highly efficient and selective reagent and catalyst in one of the major goal of research in chemistry. The science of matter and of its transformation. The particularly remarkable features displayed in this respect by the natural catalyst. The enzymes has provide major stimulus and inspiration for the development of novel catalysts by either manipulating the natural version or by trying to devise entirely artificial catalyst that would nevertheless display similar high efficiency and selectivities. In this Nobel Award lecture in 1902, Emil Fischer, that is the scientist Emil Fischer, has shown remarkable presence when he said, I can foresee a time in which physiological chemistry will not only make greater use of natural enzyme but will actually resort to creating synthetic ones. Important quote by the Emil Fischer. On enzyme. Since enzymatic reactions involve binding of and reaction with a precisely defined substrate, they have the characteristics of a supramolecular process. On the other hand, reactivity and catalysis represent major feature of the functional properties of supramolecular system. Molecular receptors bearing appropriate reactive groups in addition to binding sites may complex a substrate react with it and release the product, thus regenerating the reagents for a new cycle. We see in next figure. Supramolecular chemistry, that supramolecular reactivity and catalysis thus involve two main steps. First is the binding, and which is select the substrate, the transformation of the bound species into products within the supramolecules formed. Both steps take part in the molecular recognition of the productivity substrate and the require the correct molecule informations in the reactive receptor. Compared to molecular reactivity, a binding step is involved that proceed the reaction itself. Catalysis additionally comprise a third step, the release of the substrate. The selection of the substrate is not the only function of the binding step. In order to promote a given reaction, the binding should strengthen substrate so as to bring towards the transition state of the reaction. Thus, efficient catalyst should bind the transition state more strongly than the free solute of the substrate in order to lower the free energy activation. You can see that in the above diagram, schematic representation of the supramolecular catalysis process. That is catalyst. That is catalyst, and this is substrate. Catalyst and substrate, and this stability activity binding process to form a CS complex. C means catalyst and substrate, substrate catalyst. This reaction is adsorption reaction, not is absorption, because enzymatic or catalytic reactions are adsorption reaction. Catalysts are adsorbs the layer of the substance substrate. After that reaction, it is not stable CS complex to convert into a product. This step is rate selectivity turnover. However, the design of catalyst capable to optimal transition state stabilization does not consist in searching for stronger binding to restrict transition stage analog TSA. Transition stage analog of the substrate, but rather TSAX. This is the transition state of TSA minus X, where X represent the atoms, the functional groups in catalyst that react with the bound substrate. A major role is played by the existence of strong interaction between the substrate and the receptor site of the catalyst. They may be used to facilitate the reaction in several ways, such as thermodynamic effect, strong binding forcing the substrate into contact with the reactive group, a steric effect, fixation of the substrate being able to distort it towards its transition state geometry, an electrostatic effect constituting of a possible activation of the functional group of the catalyst by modification of their physicochemical properties. As a consequence of substrate fixation, which may perturb charge distribution in both the substrate and catalyst with respect to their free unbound state, such as activation of the catalyst by the substrate itself, is a short suicidal behavior. The substrate facilitating its own consumption. The effect induced by the fixation of the substrate play a role in the reaction catalyzed by either artificial species or by enzyme and may result in the short manual activation of the two partners. One must note that number of processes described amount to facilitating in given reaction catalyst cycle, but not to full catalysis due to the slow reaction regeneration step product inhibition. The term catalysis will be applied to example mentioned below this restriction mind. Now we discuss catalysis by the reactive macrocyclic cation receptor molecule. The ability of 18O6, this is the supramolecule 18O6 macrocycles polyether. This is macrocyclic polyether bind primary ammonium ion. Often possible to induce chemical transformation on such substrate. Activation orientation binding was observed for the hydrolysis O-acetyl hydroxylamine, which forms such as stable complex 27B. Here R is acetate with the macrocyclic tetracarboxylate receptor that it remain protonated and bound even at natural pH despite of the low pKa of free species. As a consequence, its hydrolysis is accelerated and exclusively gives accelerate and hydroxylamine, whereas in the presence of potassium ion, which is displaced of substrate, it yield also acetyl hydroxymic acid. Thus, strong binding may be sufficient for markedly accelerating reaction and affecting its course. a result that also be an enzyme catalyst reaction. The chemical transformations may be induced the reaction between a bound substrate and functional group borne by the macrocyclic receptor unit, as illustrated in that the structure number below is 74, that X is equals to carboxylate and etc. Here is R, here is A, and here is B. And this is the supramolecule also. Ester cleavage process have been frequently investigated in enzyme model studies. Macrocyclic polyether fitted with the chain bearing thiol group cleave activated ester with marked rate enhancements of chiral discrimination between optically active substrate. The optically active binaphthyl reagents in above diagram perform thiols of the activated ether of amino acids with pronounced acceleration and chiral recognition. The tetra L cysteine macrocycle binds para-nitrophenyl esters of amino acids and peptides and reacts with the bound species, releasing para-nitrophenol as in shown in next diagram. The reactions displays number 1, substrate selective in favor of dipeptide ester substrate with the second marked rate enhancement. Third is inhibition by complexes metal cation that displace the bound substance. 4 is high chiral recognition between enantiomeric dipeptide ether, and fifth is slow but definite catalytic turnover. An interesting effect was the finding that binding of the substrate probably activates the thiol groups by increasing their acidity. An illustration of the suicidal effect noted above that may also be of importance in enzymatic reaction. Hydrogen transfer has been induced with macrocyclic receptor bearing 1,4-dihydropyridyl DHP dihydropyridyl groups. Bound pyridinium substrate are reduced by hydrogen transfer form DHP side chain within the supramolecular species. The first order intracomplex reaction is inhibited and become bimolecular of displacement. The bound substrate by complex cations. Reaction with the carbonyl or sulfonium substrate have been performed with the order of DHP containing macrocycles. Now we discuss catalysis by the reactive anion receptor molecule. Reactive anion receptor molecule. There is development of anion coordinating chemistry, and anion receptor molecules has opened up the possibility to perform molecule catalysis. An anionic substrate of chemical and biochemical interest, such as adenosine triphosphate, the catalysis of the phosphoryl transfer of the particular interest, namely in view of the crucial role of such process in biology and of numerous enzymes that catalyze them. Adenosine triphosphate hydrolysis was found to be catalyzed by a number of the protonated macrocyclic polyamines, in particular 24 and 6O2, that is about 38, strongly binds ATP and markedly accelerated in hydrolysis to ADP, adenosine diphosphate, and inorganic phosphate over a wide pH range. The reaction present first order kinetic and it catalytic with turnover. It proceeds via initial formation of a complex between ATP and protonated 38, followed by an intracomplex reaction that may involve a combination of acid, electrostatic, and nucleophilic catalysis. This is the anionic Now we discuss on supramolecular metallo-catalysis. Supramolecular metallo-catalysis. Here is we use metal used in catalysis. That is, we can say that metallo-catalysis. Supramolecular metallo-catalysis can consist a principle of the combination of a recognition subunit, that subject, substrate, and metal ion bound to another subunit, that is the reactive site. Complex metal ion presenting free coordinating positions may present a variety of substrate activation and the functionalizing properties. Heterotopic co-receptor such as 70 bind simultaneously a substrate and metal ion, bridging them into the proximity, thus potentially allowing reaction between them. Now catalysis of the synthetic reaction. Supramolecules are used in the synthetic reaction also. A further step, supramolecular catalysis lies in the design system capable of the inducing bond formations rather than bond cleavage process. Thus, effect of synthetic as compared to the degradative reaction. To this end of the presence is several binding and reactive groups in essential. Such is the case of co-receptor molecule in which subunit may cooperate for substrate bind and transformation. They should be able to perform catalysis by bringing together substrate and cofactor and mediating reaction between them within the supramolecular architecture. Process this type have been realized in supramolecular phosphorylation reaction. Indeed, the same 24N6O2 macrocyclic 38 as that already used in structures of the ATP hydrolysis was found of the mediated synthetic polyphosphate from the acetyl phosphate ACP. Substrate consumption was accelerated and catalytic with more turnover following the steps. First is the substrate acyl phosphate binding by the protonated molecule catalysts. Second is the phosphorylation of above within the supramolecular complex, giving the phosphorylated intermediate PN81. Third is the binding of the substrate HPO4 to minus, P means phosphate, and fourth is phosphoryl transformation from PN to P with formation of pyrophosphate PP. In below figure we see that. And 5 is release the product end of the free catalyst for a new cycle. PP is also formed a hydrolysis of ATP in this presence of the divalent metal. Here is the explanation with the diagram. We can say that this is the regeneration, transfer, intermolecular reaction, and ligation. The figure 7 and 8 we discuss. This process was extended to phosphorylation of various substrate, in particular of the synthesis ATP and ADP mixture solvent, and is aqueous solution in the presence of magnesium plus 2 ion, probably via formation of a tertiary catalytic species. The latter abiotic ATP generating system has been coupled to be sets of ATP consumption enzyme, resulting in the production of NADH, nicotinamide adenine dihydrogen, by a combined artificial natural enzymatic process. Here is 24N6O2 acetyl hydroxy. Acetyl phosphate converted into acetyl hydroxy, and after that ATP convert into ADP, and this is step is HK. HK means hexokinase. HK means hexokinase. Glucose convert into glucose 6 phosphate, and glucose 6 phosphates are convert into 6 phosphate gluconate and 6 phosphate NADP convert into an NADP convert into NADPH. 6p gluconate are convert into ribulose. Ribulose 5-phosphate in the 6p GDH. GDH means di glucose dehydrogenase, and convert it to NADP to NADPH. Now we discuss biomolecular and abiotic catalysis. The design of the supramolecular catalysis may be made as biological material and process to try tailoring appropriate recognition sites are giving high rates and the selectivity of the reaction. Modified enzyme obtain the chemical mutation or a protein engineering. Represent biochemical approach the artificial catalysts. This is also case for the generation of the catalytic proteins by induction of antibodies. Antibodies to reactive heptanes are able to facilitate the transformation of the bound species. Generating antibodies against analogs to transition state should lead to transition state stabilize and facilitate of the process. Such catalytic antibodies or enzymes have been produced for a variety of reaction. Catalytic antibodies or enzymes and the reactive field of the research has developed along such lines. It represents an approach to substrate specific, efficient and selective catalysis to supramolecular type. That is the much basic applied interest. The strong affinity for the TS for the reaction gives substrate leads it along the way and thus facilitate the process. In line with the remark made at the beginning of this chapter, the antibodies should be generated not against a TSA itself but against TSAX isoester lacking the groups which belong to the reactive function of the protein that are expected to perform the reaction. The required designing species in which the face representing the gap be ideally chosen so as to lead to the induction in the antibody of the desired reactive functional group at the correct position. An abiotic approach much interest consists of the generation TSA impurities on the surface of the bulk solid state material. Supramolecular catalysts are by the natural abiotic chemical reagents that may perform the same overall process as enzyme without following the detailed pathway by which the enzymes actually affect them or under condition in which enzymes do not operate. Furthermore, the most significantly, this chemistry may develop system realizing process that enzymes do not perform by displaying comparable high efficiency of the selectivities. Thank you so much.